Hi all. Welcome to week 4 of Bio 230. By the way, week 4 of 5, so we're really getting close to the end here. And I first want to say that I really appreciate the effort that everyone is putting into this course. I think it's going. I don't want to say like well. I think my definition of the word well has changed over time, but I really do think that we're doing the best that we can given these really chaotic circumstances, and I appreciate the effort that everyone is putting in. So today I made this short video because I want to go over some concepts that I think are particularly important in chapter 12, which talks about eukaryotic chromosomes. So if I can go to the next slide, there we go. Okay, from the first half of the course, we saw the structure of DNA. We saw this double helix with A's and T's and G's and C's. And this was DNA that I'm going to call naked DNA. This is uncompacted, unorganized DNA. And if you took all of the DNA that is within the nucleus of a human cell and you stretched it out in this naked form, it would be about 6 feet long. And our cells are obviously not 6 feet long because my entire body isn't even 6 feet long. And so obviously something has to happen, right? You can't have that much DNA in a single cell. So that thing that has to happen is DNA compaction. That is what's going to allow that really long naked DNA to fit inside of a human cell or any cell. And so this is a quick drawing, but we're going to go over these levels of compaction that get us from this DNA into the condensed chromosomes that you can find in a cell. And if you take away nothing else from this video, if you just remember one thing, it's that the more compact the DNA is, the less genes are going to be expressed on that and that compact DNA. In some regions of chromosomes where DNA is highly compacted, you won't have any gene expression at all. The cellular machinery cannot physically access that sequence to transcribe DNA to RNA to make those proteins. So what are those levels of DNA organization? I promised you that we would go over. So on the far corner over here, you have DNA, which in isolated patches in the chromosome in the cell, you can find DNA in just that normal double helical structure, but obviously it can't be like that throughout the cell or throughout the whole genome because it would be way too big. So as we move from the left of the screen to the right of the screen, you're going to see the DNA get more and more compacted. And we're going to step through these one by one, but you can always come back to this slide if you want to figure out where we are. So the first level of organization are nucleosomes. So nucleosomes are DNA that are the DNA is wrapped around 8 histone proteins. So those 8 proteins are 2 sets of H2A, H2B, H3, and H4. And you're going to have about 160 base pairs of DNA wrapped twice around this core histone octamer because it's 8 proteins, 8 mers. And then about 40 base pairs of DNA is going to connect one nucleosome to the next, and we call this the linker DNA. And just based on this level of compaction, we can get those 6 feet of linear DNA to about 1 foot long. And this is the conformation that we saw in an earlier video and you see in your textbook. It's commonly referred to as the beads on a string because you can see there you have your nucleosomes with DNA wrapped around that kind of look like the beads, and then the linker DNA is the string. And these genes are under active transcription. You can see that's not too compact. You can maybe imagine a little RNA polymerase fitting in to transcribe those genes. The next level of organization, sometimes called the 30 nanometer fiber, is when you add another histone protein, add in histone H1. And here's a figure from your book over on the right, which is showing you how that would fit in. And this is going to allow those individual nucleosomes to form a higher order organization, and that further condenses the DNA. And as I said before, if you only remember one thing, that is that more compact DNA has less active gene expression. Now, there are a few more levels of organization, and to get to these, you just keep adding additional scaffold proteins and being pretty vague here because this is an ongoing area of research. So you have the 30 nanometer fiber, you add some scaffold proteins, and you just get to higher orders of these squiggly lines. If you want to look up some more elegant microscopic pictures of those, you're welcome to. But again, the main idea here is you keep adding proteins and you get more and more organized. And eventually you end up all the way on the right, which is a metaphase chromosome. I think we've drilled this into your head now. You can see those two sister chromatids. And that is the most compact form all the way on the right, the metaphase chromosome. And that chromosome is ready to divide. Everything is nice and packed, but those genes aren't being expressed because that cell is just dividing. So really, as you can tell from how quickly I sped through those, we know most about the nucleosomes. It's those higher order levels that we still don't know as much about. So I'm going to talk more about the nucleosomes, but you're going to have to go to graduate school and do some science to figure out more about the others because we're still learning about it. So the first thing, let's look at what happens when certain regions of DNA are wrapped up in nucleosomes. So this is a figure from your book, and you see that in panel A up at the top, you have promoters. Remember, this is where RNA polymerase binds to express genes. Here, the promoters are hidden. They're wrapped up in nucleosomes. They're inaccessible. RNA polymerase can't bind there because that region of the DNA is wrapped up in a nucleosome. So you're not going to see gene expression in that top configuration, but nucleosomes can be rearranged. And when this happens, or what we call chromatin remodeling, you see on the bottom, you can then expose the promoters. So now RNA polymerase and transcription factors can bind, and you have gene expression. You also see here in that promoter, there are these DNase hypersensitive sites. That's not a natural process of the cell. What your book is actually talking about there is a technique that we use to find these regions of the chromosomes. So what scientists do with these hypersensitive sites, DNase is exactly what it sounds like. Scientists aren't too creative. It's an enzyme that chops up DNA. So, which I've given this little blue Pac-Man here as the DNase, and we're going to look at those two in the previous slide here. You have the nucleosomes that are wrapped up, and then after chromatin remodeling. And so let's imagine that we had those two pieces of DNA before and after chromatin remodeling, and we tried to chop them up with DNase. So you can see over on the left, before chromatin remodeling, DNase physically cannot access it. That enzyme is too big. It's not going to fit in there, so it can't do any chopping. But after chromatin remodeling, on the right, that you've had those nucleosomes move apart. So now that promoter region is exposed, and that DNA is now open for chopping by DNase. And so that DNase is going to cut the DNA backbone. And what is that going to look like for us as scientists? So now what we have is over on the left, before chromatin remodeling, DNase can't cut. We're not going to get any DNA fragments from that region because that DNA was protected. It couldn't be chopped up. But over on the right, after chromatin remodeling, those nucleosomes moved apart, DNase can access that region, and it produces these DNA fragments. So next, we're going to sequence that DNA, just like we talked about last week. There are a couple of steps to do that, and we're going to map it to a reference genome. We kind of talked about this with the Nick Volker case. There is such a thing as a reference human genome, for example. And once we know our sequence, the A's and T's and G's and C's, we can match them up to where they map on a human genome. And what we would find, what you see, if that orange bar is the entire human genome, or at least this region that we're interested in, we would see that our fragments only mapped to that one region of that orange reference genome. And so we might call that our DNase hypersensitive region. And what that would suggest to scientists is that that region of the DNA is open, and it might be somewhere that regulatory elements could bind, that promoters could bind, and that would be a more active region of the genome. And if you had been following along in your book, looking at figure 12.10, you might have realized that I'd cropped out a third panel at the bottom, and I'll talk about that now. So the top two are the same, A and B, but then there's C. What you see in C is that the nucleosomes are even more tightly packed than they are in A. And this is something called chromatin. So this is just especially condensed DNA. Here, RNA polymerase and transcription factors cannot bind, sometimes ever, really. So they're not getting gene expression, and what you see here is we're calling this region silenced. So just some vocabulary we've introduced this new word, heterochromatin. It's called heterochromatin because it stains dark with these DNA binding chemicals. You can see it over here. On the right, there's a figure of human metaphase chromosomes that were stained with this special technique that's going to bind regions where it's packed more tightly. And in those chromosomes, you can see that heterochromatin region tends to be around the centromere specifically in human chromosomes. And the opposite of heterochromatin is euchromatin. So those are the regions of those chromosomes that stain light with DNA binding chemicals, and that is where the less condensed DNA is. And there are a few types of heterochromatin. So there's constitutive heterochromatin. So this is chromosomal regions that are highly condensed almost all of the time. These chromosomes contain repetitive sequences, SSRs, short simple repeats, and transposable elements or transposons. These are regions, these are little sequences of DNA that can kind of copy themselves and hop around the genome. And I think we're going to have a quiz question that's going to ask why we think constitutive heterochromatin regions of the chromosome might contain more of these repetitive, non-functional sequences. There are very few genes, actively expressed genes that you need for a cell to function in these regions, and they're almost always silent, so no expression. Constitutive heterochromatin, always heterochromatin, just not being expressed. On the other hand, facultative heterochromatin, those are chromosomal regions that are highly condensed in some cells, but euchromatic in other cells. So these regions aren't being expressed in some cells, but they are in other cells. This should start to sound like a new concept to you in genetics. And we're going to talk about this case study of position effect variegation in Drosophila eyes to think about what the weird new thing that's going on here. So position effect variegation in Drosophila is a way that scientists study heterochromatin, and specifically facultative heterochromatin that can spread. So what you're seeing is over here, there's one chromosome. This is, I think, the X chromosome in flies. And there's this region of heterochromatin around the centromere. That's the blue squiggly line. And remember, heterochromatin is not being expressed. There's a region of euchromatin, the kind of lighter blue straight line. And on this, wild type X chromosome, there are two genes, white, the wild type white gene, and then roughest, the RST, so the wild type roughest gene. And let's just look at the top for now. And if you look at that fly, then it's going to have a red eye because the wild type white gene makes red. I know everything is named backwards. But so the wild type white gene is always expressed because it's in the euchromatin region that's far away from the heterochromatin region. And it's expressed in all of the cells. So this fly has completely red eyes that have a smooth appearance. But if an inversion, when the DNA gets flipped around in a chromosome, if an inversion were to happen that put white and roughest closer to that heterochromatin region, sometimes some crazy things can happen. And just being close to heterochromatin region in a chromosome can silence the euchromatin region too, and we call this heterochromatin spreading. So what you see at the bottom in that variegated eye is that white, the wild type white gene, and notice it's not now a different allele, the DNA sequence of the white gene did not change. The DNA sequence of the roughest gene did not change. It's just that now that gene is close to the heterochromatin region. So in some cells, white is still expressed, wild type white, and these are the cells where the heterochromatin didn't spread. But in other cells, that white is silenced. So now those eyes are appearing white, and this means that in those cells, the heterochromatin did spread. And what happens is you end up with this patchy or variegated eye, so where it has some white cells and some red cells. So we know that there's something about some sort of boundary between the heterochromatin and euchromatin region, and there's this concept of boundary proteins that set that barrier, but obviously they can change. But we do know from experiments that this boundary is set and it spreads linearly along the chromosome. So what you can see here is if we look from top to bottom. So in the top, we have a fly with red and smooth sectors. So what we see here and what's being diagrammed is that the heterochromatin did not spread at all. So in those red, smooth regions of the eye, the wild type white gene is still being expressed, and the wild type roughest gene is still being expressed in those red smooth regions. Up at the top, we have sectors of eyes that are now white, but they're still smooth. And so what this means is that there was some heterochromatin spread. You can see here that that blue squiggly line now invaded and it's crossed over the white gene. So now wild type white is not being expressed, so the eyes appear white, but wild type roughest is still there, so the eyes still appear smooth. If we go to the third one, now we're looking at that bottom section where the cells look kind of bumpy and they're white. There was a lot of heterochromatin spread. Both of those genes that used to be in the euchromatic region are now in a heterochromatic region. They're not being expressed. So that cell, those cells, all of those cells at the bottom part of that eye, even though if I were to sequence them, I would see that they have wild type white and wild type roughest. But the physical appearance is the exact opposite, that they had the alternative alleles at those. So this is the transformative concept that I'm talking about. I think maybe for the first time in this course, we're seeing a case where your genotype does not match your phenotype, just looking at sequence data alone. This came up in some of the last quizzes when I asked this open-ended question of what are some scientific and ethical considerations you might have for using genome sequence data to treat human, like in medical settings. And a lot of y'all said that was a real scientific concern, is that as scientists, we're not actually that great at predicting phenotypes from genotypes. If I looked at that fly, I would have told you, oh yeah, it's definitely going to have 100% red, smooth eye cells, but obviously it doesn't. So phenotype does not always match sequence, and sometimes that's because of chromatin remodeling. Now, the very last panel of this figure is something that we never see. We never see white eyes with, red cells with a rough appearance. And what that tells us is that heterochromatin never skips. It spreads along the chromosome, or it doesn't. It doesn't just like jump from patch to patch. And this is more evidence for those boundary proteins that are setting a barrier. So I think that's all that I wanted to say with this video. I'm hoping again that you understand the relationship between DNA compaction and gene expression, and that you've started to get really curious about different ways that we might see an unexpected phenotype given the sequence genotype that we observe. So thank you.